All right, chapter 17 is one of my favorite chapters. And the reason being, when I was a little kid, I remember that in boxes of cereal, we used to get toys. And one of the toys that seemed to be the one that everybody on the neighborhood wanted, or at least all the kids in my neighborhood, was this little blue plastic watch that was a code watch. And it had the alphabet on it, and it had another plastic ring on top. And you rotated it, and one letter meant another letter. And you could write notes back and forth to your friends in school, and as long as they know how you had turned your dial to make the note, which I don't remember exactly how that was conveyed, but anyway, they could decode your message. So you could pass notes in school, and the teachers, if they caught them, it would be gibberish. Yeah, let's see. Hold on a second. Let me check the schedule a second, because I do have to get through 17 today. The review really isn't going to be a whole lot of new material. I'm not going to ask you anything about those labs we did with the Vernier software. So I'm going to ask you genetics problems, questions about genetics problems, and some of that stuff. Well, I mean, it goes into lecture. We did it in lab, what we did in lecture too, the X-linked and all that stuff. And then mitosis and meiosis, which you've already had practice studying for that because you had some of the images on the exam, remember? So yeah, and then there's going to be metric system conversion. So yeah, I think we'll have time at the end. We can spend a few minutes at least kind of talking about what's going to be on it, okay? Will that work? Sure. So the reason why I was telling you that long, drawn-out story about the watch, because I always thought that coding stuff was kind of cool, like one thing means another. So what we're going to do today in Chapter 17 is we're going to answer that question, I don't know, that you probably had in your mind since the beginning of the semester when we talked about the primary level of protein sequence, or primary level of protein structure, excuse me. So if you think about that for a second, we were back talking about proteins, and proteins have a very explicit shape. Enzymes are proteins, and they have to be shaped the same way, and you've heard me say this probably ad nauseum, right? So the thing is, if something has a specific shape, and even if to us it looks like a glob, if something has a specific shape, if every enzyme glob is supposed to be shaped the same way, then there must be some directions on how to produce that protein. That protein is a molecule. Molecules, for all their wonderfulness, certainly don't have a mind. They can't say, well, this is how we're going to come together and form this protein. And you remembered, of course, we talked about proteins. We talked about the building blocks. We talked about amino acids. And we said the way you make proteins is you link amino acids together, and there's 20 different amino acids, right? Threonine and leucine and lysine. And I think at the time, I'm sure I told you don't bother to memorize the names. But, they are those molecules, remember, that are all share one very big thing in common, and it's the fact they have two functional groups, right? They're amino acids, an amine and a carboxylic acid, or a carboxyl. It means the same thing, all right? Now, where we're putting this together is how does the cell know what the primary level of sequence structure is supposed to be? In other words, remember we talked how important it is to get those amino acids in the right order, that sometimes getting just one in the wrong order is going to, can end up with really bad side effects like sickle cell anemia, which we'll talk about later on too. Okay, so, where is that information on how to make enzyme glob B or C or A or whatever one you're trying to think about, sucrase, you name an enzyme? Well, that information is your genetic code, which I don't think there's anything new because I've kind of referenced it to you guys a few times throughout the semester, and you may have already known it. But I'm not sure you knew that it's specifically what the genetic code codes for, okay? It codes for you. It codes for your traits. It codes for being a purple or a white flower, blah, blah, all that stuff we've talked about. But what the genetic code codes for is that sequence that we talked about way back in whatever chapter that was, that primary level of sequence, that recipe on how to make a protein. How do you make protein X? Well, you start with this amino acid, then you add this one and this one and this one and so on and so forth. And if you get the recipe right, then because they're molecules, and molecules do things like attract each other and repel each other, and they attract each other in different ways. Some ways that are very strong, like covalent bonds, and some ways they're kind of weak, like hydrogen bonds. But they do these things because atoms do these things because of the laws of physics. So as a living organism, if you can get these molecules with these different interactions chemically to line up in the right order, blah, 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 they fold themselves into the perfectly shaped protein you're looking for. Right? So that's where we came from, and where we are is we're now going back to that point where we had that primary level of protein structure, that sequence of amino acids, and we're going to talk about how that happened. How did you get the right protein in the right order? How did it go from your genetic code to your protein, to whatever protein we're making? So you're going to see this again, so don't worry about looking at this too hard right now, but this is that decoder table, similar to that decoder watch that I was telling you about, but I'm going to talk a lot more about that later on. But first I want to get into the basics of this, to the central idea, to what we call up above in the red, the central dogma. Dogma is kind of a word you don't hear much, and dogma sometimes can be negative. It has negative connotations, and sometimes it has positive connotations. A dogma is, well, I guess for me, the best way for me to think about dogma is like when we say, why do we do this? Like say at work, why do we do it this way? And someone tells you, well, we've always done it that way, right? You've come across that. So, so a dogma is kind of the way things are accepted to be. They're incontrovertible truths. Really nothing's ever quite truth in life, and all that philosophy behind that, metaphysics sort of stuff. But a dogma says that, this is the way it is because we've always, or well, not always, but we figured out that this is the way it is. So you can have a bad one, you can have a good one. Now the good one, an example of a good one, is up above. And what I mean by this dogma is back in biology, back in history, before we knew specifically how this all occurred, before we filled in the arrows you see in that little flow chart, we knew that DNA controlled for a molecule called RNA, and that controlled for a protein. So in other words, that recipe, that message was being passed between the DNA and the RNA and passed to something that used it to make the protein. Okay, so that message of which amino acids go in what order. Now we knew that. We had evidence to support that. What Watson and Crick did, or I'm sorry, not what Watson and Crick did, they figured out the molecule. But what biologists figured out after that was the arrows that you see there. So specifically, okay, think about it for just a second, folks. DNA is a molecule. It can't turn to RNA and say to RNA, hey, RNA, to make enzyme X, you start with amino acid methionine, then you add a lysine, put in 2 serines, then a threonine, right? We can do that. They can't. They're molecules. So how do they communicate? They communicate with codes, don't they? They communicate with codes. They communicate with ions. They communicate, remember, they communicate with all those things that we've been talking about all semester long. So this chapter, if you will, is really sort of how the communication happens. How does that information get passed between these non-living things to create a living thing, right? It's kind of cool. Okay, now, we're going to break the two processes up, the two arrows, if you will, into two separate things. The first one's going to be called transcription, which means literally to write, and you'll see what I mean as we go through this, what we're writing. And then the second letter, or the second arrow is translation. So think about back to my decoder watch thing again, right? I was writing down my code, oh, this teacher sucks, blah, 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 whatever I was doing with my little decoder watch, passing it to my buddy, assuming it wasn't intercepted by the teacher. He was taking his decoder watch, and again, I don't remember how you would do it, but anyway, he would set it up so he could then translate my gibberish into what it really said, right? So the same thing's happening here. And in a bacterial cell, and we'll go through this back and forth a couple of times, but the processes are essentially the same. There's a few other steps. But in a bacterial cell, it's a very simple process. You take the DNA, that double helix that's in the blue, and through transcription, you make notice the RNA there. Number one, notice the M. That's important. Remember at the beginning of the semester I said there were there were 3 types of RNA. There was tRNA. There was R, and then there was that one. I mean, we talked more about RNA, but I said, all right, just so when you see it again, you know what I'm talking about. There are types of RNA. We're going to talk about that more specifics today. But the first one that we're going to introduce here is the messenger RNA. That's what the M stands for, okay? Simple. Transcription. The messenger RNA, the messenger RNA molecule, that red strip. Now, do you know why it's only one strip? In other words, why isn't it a double helix like the DNA? Why is it only one side, or only one helix? It's not really a helix, but it's a strip. Wasn't that long ago, folks. Wasn't that long ago that we talked about the differences between DNA and RNA, and there were 3 of them that I mentioned. You're going to have to remember another one today. It's RNA. And what do we know about RNA, Marie? What do we know about RNA versus DNA? It's one of those 3 differences. That's right. DNA is double-stranded, right? DNA looks like the full ladder. RNA is like if you took the ladder and cut it in half. All right. So it's single-stranded. Now, the other thing you have to remember about an RNA will come up in just a moment. So keep that in mind. Keep in mind those differences that we talked about. The sugar's different. Okay, that's fine. You're not going to have to worry about that. But the fact that it's single-stranded, which we just answered here, which is looked at here, and then the third difference, if you remember, was that the base difference. There's no thymine in RNA. It's uracil. All right, so keep that in mind. Anyway, transcription, when we make a piece of messenger RNA, it's called messenger now because it relays the message to, and now we throw in what? Chapter 6, the ribosome. The ribosome is one of the cell organelles. Now, bacterial cells have them. Eukaryotic cells have them. What does the ribosome do? I think it was a matching question, if I remember one of your exams. Well, what is the purpose, the function of a ribosome? And while you're at it, somebody else tell me, what is a polypeptide? It's not toothpaste. Anybody, what's a ribosome do? Am I just talking into hyperspace? All right, let's see who's there. Colin? Kirian? Marie, I know you're there. Sadie, you're there. Oh, well, what does a ribosome do? What's its function? What does it do in the cell? Yeah, that's what they make, right? They make polypeptides. Right. Well, in the ribosome, the RNA, the messenger RNA, the message, okay, not the RNA, folks. Look at the picture. The little red strip is still there in the ribosome, but the ribosome is reading the message, and it's creating the polypeptide, okay, like Maurice showed us, right? So that's the basic idea of what's going on in here. The only thing we're doing for the rest of the chapter, to be honest with you, is getting into the details how that happens. All right, it's very simple in bacterial cells. You get transcription, you get translation, we're off and running. Now, in eukaryotic cells, you got to remember chapter whatever it was again. Eukaryotic cells and prokaryotic cells, or bacterial cells, differ in one really big way. Eukaryotic cells have a nucleus, okay? So that means that the DNA, which always resides in the nucleus, unless the nucleus disappears like it does in mitosis and meiosis, has to somehow get that information, that messenger RNA molecule. Don't worry, I'll tell you in a second what the pre and all that's about. but it's got to get it out of the nucleus through one of those pores out to a ribosome, because it's very simple, folks. The DNA stays in the nucleus, the ribosome is in the cytoplasm. One is not in the same place as the other. Now, the DNA stays in the nucleus is a good thing, because the nucleus is like, remember, another level of protection. Those nuclear pores that you see the messenger RNA going through, they just don't let anything through. And DNA, you don't want that to get messed with. If you want to know why it's not such a big deal in bacterial cells as it are in our cells, then take my Bio 2 class. Now, what is all this pre-mRNA and mRNA? Notice that bacterial cells, for the most part, some do, but for the most part, don't have that stuff. I've been talking all semester long, or at least recently I have, about this stuff that we call junk DNA. And I've told you that's not the best word for it for a variety of reasons, but you understand what I mean. It's basically DNA that is not turned into a protein. It's basically information on the DNA that can't be used to make a polypeptide. So years ago, we're like, why is that there? We just called it junk, but we're learning more and more. All right, now, when you make a piece of messenger RNA, when you transcribe a piece of messenger RNA, you transcribe all the DNA. You transcribe all the parts of the good parts. You transcribe all the parts of the bad parts, the junk parts. All of it gets transcribed. It's like the postman, when he comes to your house, gives you all the mail, all the stuff you want, and all the crap you throw right in the garbage. He gives it all. It's up to you to figure that out. Now, the reason why this doesn't happen in most bacterial cells, again, not all of them, but most of them, is because they don't have this junk DNA. They have a much simpler genetic makeup. And that's enough for that right now, okay? Back to the eukaryotic cell. So what we're doing in RNA processing, that extra step, is we're taking that junk DNA out of that piece of pre-messenger RNA. Notice it's shorter from there to there. So we literally have cut those pieces out that are junk. It's like you throwing your junk mail into the garbage. And I'll talk about what the yellow parts are as we get into the detail of that. But that among other things, that helps the messenger RNA find a ribosome and be protected and actually make the trip out there to the ribosome. All right, so once that RNA has been processed in eukaryotic cells, then it can leave the nucleus and go out about its business. And for all intents and purposes, it's the same as it was in the bacterial cell, right? Anybody got any questions on that? Now, no matter what we do this entire chapter, except toward the end when I talk about some mutations, but as far as this process, this is what I'm talking. This is the forest, folks. This is what we're going to do today. We're going to talk about how this all happens. So does anybody have any questions? We're all good? All right. Now, let's go and talk about the code part first. All right. We'll talk about the code part. Then we'll kind of put the code part with the physical part. So let's go and just really drill down to a piece of DNA. Is this in your book? It might be 17.4. I can't remember. I have several pictures that I mix up in this sort of stuff. Anyway, not to confuse you, but just because I like them better. Back up at the top, there's the DNA. Your typical, everyday, run-of-the-mill, very, standard double helix, double-stranded. They've unwound it for you, so it's a little easier to look. And you should definitely recognize all those letters, right? That's the code. A, C, A, T. Those are adenines and thymines and cytosines and guanines. Now, you should also remember that DNA is anti-parallel, right? That's important for a number of reasons. It's important for explaining, what the Okazaki fragments that we talked about when it was being copied. It's also important for understanding that DNA has one template strand. In other words, only one of the strands contains the code. Now, I understand if you're looking at going, wait a minute, the other side does too. Yes, but in opposite. So think about the DNA template strand as being the photograph that you look at of mom and dad. Nobody gets those anymore like that. But in the olden days, right? And the other strand would be the negative. Nobody puts the negative up on the wall and says, what the hell is that? That's mom and dad in a negative, So the template strand is the strand that's red. And notice the direction in which it's red. Remember back in replication? The reason why the leading strand can be leading is because it's reading already from the 3 to the 5 prime. All right, so the DNA template strand is going to be read during transcription, the same way it's read during replication from the 3 to the 5. So this strand at top is going to get read. The other one is not. The other one is basically, protective redundancy. Protective redundancy. And let's say something happens to the DNA and this cytosine gets messed up. You know, here comes an ultraviolet ray because you're laying at the beach long and destroys that thing. Well, those things that can repair your DNA go along and they go, oh, I know it goes there. Cytosine does, because on the other side is guanine, right? And also stabilizes the molecule. All right, now messenger RNA is temporary. That piece of paper I passed to my buddy was meant to be thrown in the garbage, back to my decoder ring. So messenger RNA can get away with being single-stranded because, like I said, it's not worried about long-term preservation. Now, it does get messed up, so we'll talk about that, but usually it does not. All right, so back into transcription. What we're going to do in transcription is Chargaff's rule with a modification. Now, y'all remember Chargaff. Chargaff's the guy that figured out A equals T and G equals C. In concentration, in proportion. I showed you an example in class of how you could do that. But basically, adenine, what he's saying chemically, adenine binds to thymine and guanine to cytosine. There is one exception, right? And that's that final difference between RNA and DNA. The sugar, the strandedness. Come on, folks, what's the last difference again between DNA and RNA? It's very important at this point right here. If you look at the screen, you can see it. Great. So if I show you a piece of DNA template strand on the exam, I was going to cover with my hands. Holy cow! So in other words, if I cover this bottom part, if I covered all this and I just gave you this up here, and then maybe I also gave you a sequence that looked like this, and I said, okay, which one is RNA? So in other words, I only gave you half of a piece of DNA, so you can't go with the strand in this stuff, right? I don't show you the sugars. So the only thing that's left to do is recognize that in RNA, wherever you're tempted, let's say, to put a T, it's a U. All right, so follow the template strand and see if you don't come up with this messenger RNA strand. Give you a second. What I'm saying is do the Chargaff's rule thing and see if you don't come up with that. It's good practice. You might see something like that on an exam, so I might have you do that on an exam. Does everybody come up with the letters in the messenger RNA that you see down below? Did you all come up with UG, U, GUSK, KUKA? UGG, UUU. Everybody good with that? Give me a thumbs up, a yes or no, a head nod. All right, good deal. All right, now, out in the ribosome, here's the next part of the code being transferred or being deciphered, if you will, at this point, being translated, right? In the ribosome reads the piece of messenger RNA, okay? And it reads it 3 bases at a time. A base is a U, a G, an A. Even back in R and DNA, a base is a T. All those letters are bases. So in the ribosome, chemically speaking, when the ribosome comes across the codon, and codon sounds just like code for a very good reason, it knows that UGG codes for the particular amino acid. In this case, it's tryptophan. Again, don't worry about the memorizing the names of the amino acids, but you can see quite clearly that's TRP. It then reads the next codon, reads them 3 at a time, UUU. The ribosome says, oh, that's phenylalanine. GGC is glycine, UCA is serine, Guess what that is at the bottom, right? Guess what this is? This is the primary level of sequence, isn't it? That's the primary level of sequence. Now, if you get that sequence right, because of all the, you remember, amino acids are all the same in the amino and the acid part, but they differ in the side groups. So you got all these side groups hanging off, interacting chemically with each other, and guess what you get? You get a blob. You get a blob of the right shape. All right, so that's the code part of this. Now, if we can break the code, if we have the little decipher ring, and we can figure out what each codon means, and by means, I mean which amino acid it is, then we can answer one of the very basic questions about life. I can look at your proteins and go back and figure out what your DNA looks like. And although we don't really talk about it in this class, in Bio 2, we talk a lot about when you're looking at what makes organisms different. Okay, what makes very closely related organisms different, like say cats or dogs, wolves, that kind of stuff, has to do with DNA. It has to do with the differences in the DNA. What makes you and I different are differences in the DNA. You see how important this is then? So knowing that primary level of sequence, we can actually trace back as long as we know what the codons code for. And guess what? You saw it already. We do. You see? It's a code table. Now, there are a variety of permutations. I showed you one at the beginning that was maybe a little more colorful, but it works the same way. Read the outside of the table. Along the left, what you're doing is you're figuring out what codon stands for which amino acid. So the first base is here, second one is in that column, and the third one is broken down on the right side. So if we were to say, example, want to look at CAC, the codon CAC, what does that code for? Well, we go C, A, C, and we'd follow that, we'd find out, oh, it codes for this one. Codes for histidine. Again, I don't care if you memorize the names of them. That codes for that amino acid. Now, if you're wondering what all the brackets mean, we'll get to that in just a second. Okay, if you wonder what the green and the red ones mean, we'll talk about that in just a second, although it's probably pretty obvious, to be honest. But let's practice this a couple times. All right, folks, AAA, the codon AAA. Which amino acid? I'll erase my scribbles. AAA. Which amino acid does that code for? So if you're a ribosome, and that's the codon you're reading on the messenger RNA, you're going to know which ribosome. Or I'm sorry, which amino acid. Beautiful. Everybody see how she got that? Anybody not understand how AAA stands for lysine? You will need to know how to do this. Now, I understand it's not hard, right? But do practice it a couple of times so that during the exam, when you're a little bit nervous, and if you're looking at a table like this, you don't make a mistake, because that would be, that would be one of those mistakes that you would hate if you looked at later on at your exam. Okay, what do the brackets mean? Well, notice this word over here on the left, wobbly. You got three bases there. Mother Nature understands that things aren't perfect. She always builds in a little bit of a fudge factor. She always builds a little bit of a, what do you want to call it? She always builds a little bit of a safety net, okay? So notice that when you look through the code, as long as you get the first two letters right, the first two bases right, it usually, not always, but it usually doesn't matter if the last one gets messed up. Look at CUU, CUC. Look at these guys. Look at all those. They all code for the same amino acid. So that last base can wobble. It can change. It can vary. Now, there are exceptions, right? Like the one we did, histidine. Notice that in histidine, as long as you get a U or C in the last one, you're going to get that amino acid. But if it's an A or a G, you're going to get glutamic. Or glutamine, excuse me. Right? So that is really nothing more than Mother Nature playing the odds, and through evolution and through time, it being favored that the code be a little more forgiving than if you get it wrong every time you get a different amino acid, right? So that, remember we talked about that's a problem when you get the different amino acids. Okay, back to the green and the red ones. AUG down at the bottom. MET is methionine. It's an amino acid. There is no amino acid called start, but this is quite clearly the start codon. Just like, I was going to say, just like your CD. Nobody has CDs anymore. But just like CDs knew when the tracks switched over, like if you had an old CD player, it would say track 2, track 3. There was a code in there that said, okay, that song ended and the next one's beginning. Mother Nature is very conservative. When she makes a piece of messenger RNA, she might actually create a piece of messenger RNA that codes for 5 or 10 proteins. So if this is my piece of messenger RNA, she just inserts start, and then there are 3 stop, notice in the red, stop codons. So you get a start, stop, right? Says, okay, protein 1, protein 2, protein 3. Clever, huh? Mother Nature's no dummy. She knows and saves time that way. She can crank out a lot of protein product with just one piece of messenger RNA. Don't memorize this table. Know how to read it. That's all I ask. Okay? Questions? Okay, now we crank the screws down a little bit tighter in detail. Let's look at how we get this whole process of transcription started. In other words, how do molecules, which don't have an ability to communicate, talk, obviously, or wave their hands, how do they get this whole thing going? How do we make that, and it's very profound to me, and if you think about it, it's very profound in general. How do we go from the chemical, the physical, to the biological? There's something very different about the chemical reactions that occur in you and I and all other living things that occur in a rock, okay? I'm sorry, nothing against rocks, but come on now, when it comes right down to chemical dances, and there's some pretty good ones out there in the chemical world, if you think about like the hydrologic and the geochemical cycles, this whole one we call life is actually pretty cool too. All right, so let's go down to transcription. Let's orient ourselves. What we're doing here is we got the blue is the DNA, and the transcription unit is a gene. All right, so we're going to read a gene. And we're going to read a gene with actually something that you may have seen if you've ever gone to a concert. Mother Nature uses a guitar pick. See? No, that's actually not really a guitar pick, but it always looked like a guitar pick to me. But that is actually, that thing that looks like a guitar pick is an enzyme. And you might recognize part of the name of it. This is RNA polymerase. RNA polymerase does what all enzymes do. It does, or many enzymes do. It does what its name says. It is an enzyme because it ends in ASE, and what it does is it makes polymers of RNA. Just like DNA polymerase in replication help make copies of DNA, RNA and RNA polymerase helps mix RNA. So RNA is what is responsible for doing the transcripting process. It's responsible for reading the template strand, okay, reading the bottom strand of those two, reading from 3 to 5, so it's going to be moving this way, reading that, see, and creating a piece of messenger RNA. And it's going to read all the way down that transcription unit until the code comes and says, okay, that's done. You're done reading that. Now, as I said earlier, that's the reason why in eukaryotic cells you have to process this stuff. This piece of messenger RNA that's being created out of this piece of DNA up here is going to read all of it. All the good, maybe this is the good, mixed in with all the junk, all the good. It's going to read every bit of it. So this piece of completed RNA right here, this complete, actually we should add an M there and say this is messenger RNA, is not been processed. If it's in a eukaryotic cell, this is where prokaryotic cell, we're done. Prokaryotic cells don't have to, there's no junk parts. Okay? Now, back to that link in chemistry and biology, and also it's kind of a funny story. How does the guitar pick, how's the RNA polymerase know where to jump on the DNA? All this is happening in the nucleus. How does it know where the gene starts, right? I mean, it's a molecule. You're driving down the road, a freeway. Where does it start? So you got to have a place for it to start. Got to have an on-ramp. Oh, a few years back there, my wife and I were driving around, and the bumper sticker that says "Save the Tatas," I guess it's been around for a while. I don't know. Sometimes my wife claims that I'm oblivious to things that have been around for quite a while. But I looked up ahead at a car, and I saw that bumper sticker, "Save the Tatas," and I told my wife, I said, "That's cool." She goes, "What, the bumper sticker?" And I said, "Yeah, there's a There must be a lot more interest in genetic engineering going on now. And she goes, what? I said, Tata. There's these things in biology called Tata boxes. See them? Anyway, the Tata box, and I'll come back to my story here and finish what, what my wife finally, well, she just called me an idiot, obviously, because she told me, I guess I should have noticed there was a pink ribbon on it. They were talking about those Tatas. But as a biologist, immediately I went to, oh, Tatas. In biology, everybody knows about Tata boxes. In biology, the Tata box is called that because it's just a repeating sequence of those letters. Thymine, adenine, thymine, adenine, thymine, adenine, blah, 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 blah. It's called the Tata box, okay? A Tata box is about 25 base pairs long. Now, it starts with the ta-ta-ta, and then it ends with a bunch of other stuff. But at the end of that is the starting point. The promoter sequence of the DNA is like the on-ramp onto the freeway. You get it? That's where the RNA polymerase knows to bind. Now, interestingly enough, RNA polymerase doesn't recognize the Tata box, but these things called transcription factors do. They stick to it. Okay, so they will only stick to a Tata box. They won't stick to anything else. They're proteins. They're color purple, just to keep that color purple going throughout the theme of the chapter, right? So see how stupid I was? Tata boxes. My wife's going, oh my God. She goes, you need to get your head out of biology sometimes. All right, so now, once the transcription factors bind, what we say in biology, or what we say in chemistry really, is that when the transcription factors bind, they bind very readily to the Tata box. That actually encourages that enzyme, RNA polymerase II, okay, it's got a special name to it. It's that guitar pick. It encourages it to bind to the DNA. So it encourages it to bind to the DNA, and then once it binds to the DNA, then we can start reading it. So it's all a chemical act. You know, because you have TA in the right place, the transcription factors bind to it that way. That increases the chance. Because remember, these molecules come together and do their binding the same way or under the same rules that enzymes and substrates do their binding. By random, they bump into one another. But if you increase the chance of that happening, then you get a better chance of that RNA polymerase binding, and boom, you're off and running, right? So once you get binding, then you get this. Now, the colors have changed. This is, pretty sure this might be out of your book. I don't remember. Anyway, RNA polymerase is reading to the right. The DNA, and there are other enzymes involved, just like in DNA, making copies of DNA that help unwind the DNA so it doesn't kink up. You know, there are those that are unzipping it and zipping it back together. But it's kind of like a boat moving through the water, right? The DNA is unwinding in front of it. The RNA polymerase is doing the job you see there. It's taking these RNA nucleotides. Since RNA is just the message, once the message is been translated, then the RNA can then be chopped back up into those letters and recycled back into the nucleus, you see. It's kind of like once you use a letter in the alphabet, oh, by the way, new proclamation, nobody can use the letter B anymore because Bob in Cleveland used the letter B, right? You can use the letters over and over again. It's like a buddy of mine used to say, if you read the dictionary, you've read every book in the world because it contains all the letters. Well, I guess all the English books in the world. All right, so those RNA nucleotides are being recycled, and in us eukaryotes, we crank through the DNA at about 40 nucleotides a second. So that's how fast that guitar pick thing can move, about 40 nucleotides a second. Now, notice before we leave this slide, so I'm going to make one more emphasis. This is the template strand, and it's the template strand. Don't get confused by the double helix. It's because we're reading from the 3 to the 5, right? That's the 3 to the 5. Now, you remember I was saying about Mother Nature being this classic don't waste time or recycler, and I did in other parts of the semester, I'm sure I have, but if you look at pictures of DNA of chromosomes that are being transcribed, we see these little things called puffs, chromosomal puffs. There, you can see them in that picture on the right especially. The picture on the left is a little higher magnification, probably electron microscope. But anyway, what you're looking at here, this is the DNA, and all those little dark spots on there are RNA polymerase. So what we're saying is RNA polymerase can read several genes simultaneously. You don't have to read just one gene on one chromosome. You can do that on several chromosomes at the same time. It's all about efficiency. Now, the difference in these two pictures, folks, is just magnification. Okay? In the puff, you don't see the RNA polymerase, but you do see its effect. You see that widening of the DNA, don't you? Like a boat going through the water. I love those kind of pictures. That's cool. Okay. Any questions on that? So we've read a piece of messenger RNA, and if we're a bacterial cell, most bacterial cells, we're off and running. We're ready to translate. But remember, in eukaryotic cells, we're not quite done yet, right? Because the messenger RNA that was read was not processed. It was pre-messenger RNA, because when you do transcription, you read all of it. So what we're going to talk about now is this idea of how eukaryotic cells, how cells like you and I, go through this process of getting rid of those junk parts and processing that RNA. Put those yellow tips that I showed you in the picture earlier. All right, let's do the yellow tip parts first. At one end, what we get is we get a modified guanine. Okay, it's like adenine, cytosine. It's guanine with 3 phosphates. So it's like ATP, but it's a GTP, okay? At the other end, what we do is we add a poly A tail. And poly A tail means it's just a bunch of A's. From 50 to up to, what, say 250? At the other end. Now, what these two do, the 5' cap, notice it's called the 5' cap because it caps the 5' end, and the poly A tail are basically these three things. They help it get out of the nucleus. Enzymes, hydrolytic enzymes, remember enzymes in your cell, again, are molecules. They don't have any way of communicating. So hydrolytic enzymes that go in chewing molecules won't know not to eat up a piece of messenger RNA. So these little caps on the end help them kind of protect the messenger RNA from these enzymes trying to digest it. Even though they're existing there for good reasons, you know, they don't know. Hey, wait a minute, I'm a piece of messenger RNA. The cap on the end, the 5' cap, also helps the ribosome when it needs to attach to a piece of messenger RNA, or actually I should say that the other way around. It helps make the ribosome and the messenger RNA attract one another, stick together. Now there's also something at the other end called a polyadenylation signal. Okay, that's part of the protection for the messenger RNA. Now, as far as where our code is, as far as where our gene lies between the start and the stop codons. Okay? But notice in the picture, we got different colors of red. That's because mixed in amongst those start and stop codons, there are the junk parts. Remember, this is the pre-messenger part of RNA. Now, the parts that are light yellow are known as introns, and the other parts are known as exons. Now, those words stand for something more than just introns and exons. Introns are the intervals, and exons are expressed. So exons are the genetic code. They're the code, and introns are the junk parts. Okay, they're the intervals between. Remember I was showing you chromosomal puffs? We got 3 genes here. 1, 2, 3, divided up by these introns. So it's not enough necessarily to have a stop codon at the end of a gene. You also have these introns that divide up genes. Okay? Now, what's going to happen in messenger RNA processing in the next step is something's going to go along, this really cool thing called a spliceosome. It's this really just incredibly complex molecular machinery that actually goes along and recognizes the introns and cuts them out. Right? There's a spliceosome. See what it does? It actually takes the entire piece of messenger RNA and wraps it up in a loop, an intron loop. See, the introns again are the light red. It actually brings those pieces together. A spliceosome has little sticky parts of messenger RNA that know where to grab the introns. See that? pulls it together, and snips it out. So that works all the way up and down that piece of messenger RNA until all of the introns are removed. And then other enzymes are responsible for sticking these things back together. So now we get a shorter piece of messenger RNA, right? But it's been processed. It has no more junk. Without the junk mail, you get less mail. Don't have to do this in prokaryotes, though. One more time, right? Prokaryotes don't have the introns, although there are some exceptions. Most prokaryotes. Okay, now, where do we go from here? So we've got transcription. We've taken the piece of messenger RNA. We've cut out the junk parts. We've processed it. So let's kind of just stand back for a second and see where we're going and where we've been, right? So in a piece of DNA, there are exons chopped up by introns. Exon in the picture, exon 1, 2, and 3. And when we were talking about protein structure, we didn't talk about this level of protein structure, but beyond those primary, secondary, and tertiary levels, there are also levels of protein structure known as domains. See, we're finding out, this is so incredible, we're finding out that proteins, really big proteins, can actually act like, little towns, right? With domains being subdivisions, if you will, in my analogy here. So a protein might be responsible for taking care of a really big biological function, and it's one protein, but it's broken up into like the shipping department and the manufacturing department and the design department. You see what I mean? Now, they're all designing, yeah, they're all making, but they all make different parts of the protein. It's incredible. Proteins are almost, proteins are almost living things unto themselves sometimes. And great for code. Okay, so you get the connection there between what a domain is. See, the colors are staying the same. A domain is, and introns aren't going to have domains because the introns are going to be gone by the time you're making this polypeptide. Okay, so this is where you are at the mole picture. You guys ready to move on to translation? That's transcription. All right, so let me remind you where we are. If we're in a eukaryotic cell, where we are is we're now out in the cytoplasm. The messenger RNA has been processed. It's made its way through the nuclear pore. It's attached itself because of that 5' cap. It's attached itself to a ribosome, and we're off and running. We're going to start translating. Now, I know this picture is not in your book. Again, if I'm wrong, I'm sorry, because I think I did borrow this from another book. But anyway. You'll see what it is in just a minute. Now, in the background, the big brown thing is the ribosome. You recognize the red thing is the messenger RNA. And hopefully you recognize the little purple things floating out in the cytoplasm, because that's where this is. This is the cytoplasm as amino acids. That's why they're different shapes, round and square and 6-sided things and all that. Okay? All right, so these are different amino acids. Notice what's happening in the ribosome. Now, this picture that you see is as if you walked halfway through a movie. In other words, it's been going on for a while. It's been going on for 1, 2, 3, 4, 5, 6, 7 amino acids. So 7 steps have occurred. We'll talk in just a minute. It's called elongation. So 7 elongation steps have occurred. What we are witnessing here is the addition of the 8th amino acid, this one right here. Okay? Again, don't worry about the abbreviations. So here's what's happening. Now, if you look at it too, if on the right is where the new amino acid is coming in, notice on the left that we have one of these green-shaped molecules that we'll talk about in just a second that's leaving. Now, what those green-shaped molecules are is they are transfer RNA. That's the other type of RNA that we talked about. And transfer RNA does what it says, just like messenger RNA does. Transfer RNA brings the amino acids to the ribosome to be assembled into a polypeptide. Okay, now, there's a question in that, right? The ribosome knows what amino acid because of the codon that reads off of the messenger RNA. How does a transfer RNA molecule know that it has the right amino acid? Transfer RNA molecules, as we'll learn in just a second, are specific for the amino acid they carry. So while all transfer RNA molecules carry amino acids, only certain ones carry glycine, only certain ones carry phenylalanine. I mean, they're specific. Now, the reason why that is If you look down below what's happening in the red arrow, you'll see exactly why that is. Codons are on the messenger RNA. These are molecules that don't have the ability to think. So how does one of those transfer RNA, those blue-green molecules, know to bind? How does it know to click into that spot? It has an anticodon. Now, the anticodon is the opposite of the codon. If you think about it, the anticodon is the DNA. Replacing the U's with a T, you're actually looking at the DNA, because remember, the codon is the opposite of the DNA, and the anticodon is the opposite of that. Well, an opposite of an opposite is, right, negative of a negative. So we'll talk in just a minute how the transfer RNA for glycine with the particular anticodon CCG. In other words, the anticodon on the transfer RNA is going to decide what amino acid that transfer RNA grabs. I'll show you in just a second how that happens, although you might have a hint. It has to do with enzymes again, right? Now, the transfer RNA molecule on the left is falling out. It's delivered its amino acid. It delivered tryptophan, actually. And now it's going out, and it's going to attach itself to another, presumably another tryptophan. The amino acid, the transfer RNA in the middle is delivering this amino acid. I like to use an analogy of a doctor's waiting room. To me, the room on the left, that's where you're paying the bill, right? Or maybe you're making your next appointment. Here you are in the treatment room. Here you are in the waiting room. And you're moving along. And all along, what is happening is the ribosome is actually moving to the left. I say that because it's not the messenger RNA moving to the right. The ribosome jumps to the left. And every time it, I'm sorry, not jumps to the left, it jumps to the right, excuse me. Got that backwards in my head. It jumps to the right. And every time it jumps to the right, those codons that are in the position on the right become in the middle. The ones in the middle end up on the left. You see what I mean, right? So the codons are going to move this way as the ribosome jumps that way. Okay, let's look at this transfer RNA molecule a little bit more closely. Oh, let me mention one more piece of RNA before we go on. The last one is the rRNA. rRNA is known as ribosomal RNA. And this is the only thing I'm going to say about it because we don't really talk about it. Ribosomal RNA informs the cell or has information on how to make a ribosome. All right, so that's its job. It makes the factory, it makes that brown thing in the picture. Okay, so we've got mRNA, tRNA, and now you guys know about rRNA. Let's go back to tRNA. Take a little bit closer picture, Abbott. See, it is RNA, right? It's got adenine, cytosines, guanines. It looks like it's got 3 little cul-de-sacs, but the important parts are down below the anticodon, which is going to determine which amino acid could attach to the other site. All right, and through the picture here, folks, what we're doing is we're just showing you different images of the same molecule. See, on the right, we make it very simple. We just show you 2 parts, a little wick at the top that is the attachment site and the anticodon. All the other parts that you see, they're important for the function of the transfer RNA molecule, but not for the parts that we're going to talk about, not for the processes that we talk about. Okay, now aren't you excited to figure out how a transfer RNA molecule knows to put the right amino acid up here? I mean, that's something I would want to know. I'm like, it's a molecule. How does transfer RNA molecule floating around with all these different bases, with these in particular, AAG, know to only let a particular amino acid bind to the attachment site? Because think about it. Let me back up for just a second. Yeah. If a transfer RNA molecule brought in any amino acid, regardless of the anticodon, you wouldn't get any specificity, would you? You wouldn't get a primary level of protein sequence. You know, you got to keep that specificity all the way throughout. You got to say this part of the DNA codes for one type of amino acid, or in the wobbly bases. You see what I mean? A piece of DNA codes for an amino acid. If it's any amino acid, you lose that primary level of sequence. This whole thing is useless at that point. Well, if you remember what makes things specific in biology in terms of chemistry is fitting together. So maybe you guessed it. The way we get the right transfer RNA, what are we looking at? AUA is the anticodon. The way we get it to bind to tyrosine It's just one of the amino acids, and only tyrosine is an enzyme. And that enzyme, guess what, has a shape. And that shape only allows this type of transfer RNA to bind and this type of amino acid to bind. And then the picture on the right. Now, the name of this enzyme is really kind of cool. It's called amino acyl tRNA synthase. So it synthesizes this process of sticking the right amino acid to the right transfer RNA. Is that cool or what? I mean, that is pretty neat, isn't it? All done with shape. All right, so we're still going to be sticking with translation. We've got a few more slides to go. I need to add a little bit more detail, so hopefully you guys can get a big picture. So up above is a computer model of a functioning ribosome. The pictures down below are the same pictures, just with a little different view, right? A little more schematic type of view. So let's identify those sites that we were talking about first. The exit site, where you pay after you're done with the doctors, called the exit site, it's called the E site. The P site is the peptide tRNA binding site. The reason why it's called that is because there's a tunnel at the top, an exit tunnel, because the growing polypeptide is going to come out of that tunnel. So it's in that P site where that peptide is being made. The A site is named that because that's where that enzyme binds, amino acetyl tRNA, right? The right transfer RNA molecule, I'm sorry, not the enzyme, excuse me, but the correct transfer RNA molecule that's been made that way because of the enzyme. So it's not the enzyme that binds, it's the tRNA. Excuse me, I misspoke there. So if you see, go from the picture from the left to the right, you can see how that all fits in. Okay, so transfer RNA molecules, after they've been bound to the correct amino acid by that amino acetyl synthase enzyme, are then going to be able to bind to the A site. When they bind to the A site, the ribosome, again, is going to jump to the right. So whoever's in the A site becomes in the P site, who's ever in the P site becomes in the E site, and the process moves to the right. All along, adding this amino acid, I meant to draw it to the last one, to the circular one, right? Through that process, through dehydration synthesis. Oh my goodness, I almost said it backwards. That process by which polymers become monomers, right? Okay, let's take one more step into the detail level, and then we'll finally put this all together. Let's talk how this all starts. Now, ribosomes aren't sitting around empty with nothing to do. Mother Nature's great about, if you don't have something to do, don't sit around and find something to do. So ribosomes have actually two parts. They have what's called a small ribosomal subunit and a large, for reasons you can see in the picture that are pretty obvious. Those units don't come together to form a ribosome until a transfer RNA molecule, a special type of transfer RNA molecule, starts the process. Now, if you look at the picture on the left, you'll see what I mean by a special type of transfer RNA molecule. It's called an initiator. And the reason why it's called an initiator is, you guessed it, it's got the right anticodon. See the picture up here? To bind to the start codon. Remember, the start codon is AUG. There are 3 stop codons, 1 start codon. And notice, too, that the amino acid is methionine. That's the first amino acid. Now, there's an mRNA binding site that facilitates the molecule, the mRNA sliding into the ribosome. But once you get all this binding occurring like that, once you get the initiator transfer RNA, the messenger RNA, the small piece of the ribosome, you add some energy, because this is a reaction that requires energy. GTP, remember, is a relative of ATP. It's the same thing. And you get yourself your ribosome ready to go. Notice where the first methionine starts. It starts in the P site. Okay, it starts in the site by which that polypeptide is going to grow out of that tunnel at the top. All right, so we start there. We go through elongation. Right? You're going this way. Notice along the way, this is something that costs, right? There's energy involved. So the codon is recognized in the A site. You form the little red arrow in number 2, the dehydration synthesis that sticks the amino acid that's represented by the circle to the one that's represented by the diamond. Okay? Now, down at the bottom, this is just a split second, because right after that, the ribosome is going to jump. And when it jumps, that which was in the P site is in the E site. It exits. And that which was in the A site is now in the P site. And the process continues. Round and around and around, right? Now, what's going to stop this? What's going to stop this process? What's going to tell the ribosome, okay, we're done? Something go tap it on the shoulder. Aura, what's going to stop this? What do you think the ribosome is going to come across in the messenger RNA that's going to say, okay, done? Or Colin, or Marie, Kieran, anybody? Hello? Did you guys eat your Thanksgiving turkey too early? Everybody sleeping already? All right, I know you guys are sitting on the edge of your seats to hear about this, but there are, remember, stop codons. When the ribosome hits a stop codon, something called a release factor, not a transfer RNA, but a release factor binds to the stop codon, and guess what happens? Everything gets released. The ribosome falls apart. The protein goes off and becomes a free polypeptide, where it then folds and do all that stuff it's going to do. Notice once again, there is some energy involved in doing all this, and the release factor falls apart as well. Now, prokaryotes are actually very efficient at doing this. They can be more efficient than eukaryotes can be because they don't have that that messenger RNA processing. So in prokaryotes, look at this picture, you can actually see what's going on is the RNA polymerase is reading the DNA, and the ribosomes are reading the RNA right after that. So you're getting a bunch of RNA polymerases moving down this piece of DNA, simultaneously creating lots of protein. See, they don't have to do the processing. That's an advantage they have. So as soon as they transcribe it, boom, they're translating it. Eukaryotes, we don't have that advantage, of course. All right, last picture here is basically for your benefit. Take a look at that. This is a good picture to have in your head, to be able to describe in your head about what's going on during transcription and translation. All I've done throughout this, the chapter today is fill in the details that you see here, but this is a great kind of a stand back picture for eukaryotic transcription. So keep that in mind as you're studying this stuff. Okay, a couple of more slides. Let's talk about some effect on this protein structure when it comes to mutations. There are types of mutations that occur. One type of mutation in terms of DNA is called a point mutation, and it's called a point mutation because you can point to it. It occurs at a point. Whenever in biology we discuss something called the wild type, this is hemoglobin, the protein that makes up your red blood cells. The wild type is the normal. That's the way it is, unmutated, if you will, okay? And if you look, you'll see that the second part of the DNA template strand is a thymine, which codes for an adenine, which codes for glutamine. In those with sickle cell anemia, at a point, I've said this to you guys before, I said we're all like one letter in DNA away from having a disease. If instead of a thymine, it's an adenine, then you get a uracil, and you don't get glutamine, you get valine. That's a different amino acid. Now remember, I said the primary level of protein structure is important, but that Mother Nature does have safety nets. There are the wobbly bases. There's the fact that there are a group of amino acids that are hydrophobic. There's a group that are hydrophilic. So if you replaced one hydrophobic with another hydrophobic amino acid, even though it's not the same, you might not get the same dramatic effect. But the problem you have here between glutamine and valine, those two amino acids that get swapped, is glutamine up at top. Oops, grabbed the wrong pen. right here is polar, okay, and valine is nonpolar. So the difference you get here is you're exchanging a hydrophilic polar molecule, which would be attracted to water, for a hydrophobic nonpolar molecule, which is repelled by water. And oil and water aren't the same thing. So if you have sickle cell anemia, what's going to happen, your beta hemoglobin, remember, hemoglobin is one of those complex proteins with four separate proteins. It's that quaternary protein. There's 2 alpha and 2 beta proteins. And what happens is your beta proteins actually end up having sticky ends, you see, and they'll form, when they get together in your bloodstream, fibrous, they'll form together these sickle-shaped red blood cells, which is actually red blood cells sticking together. The hemoglobin sticks together in the red blood cells, and the red blood cells can then stick together. Now, that's not good because that clots up things, causing tissue to starve downstream. It's never good for your blood to be lumpy. It's supposed to be liquid. Now, folks with sickle cell anemia can have various levels. You can be completely free of it. You could be sickle cell anemia trait, and those folks are usually okay as long as they aren't subjected to thin atmospheres. Athletes who have sickle cell anemia trait, Ryan Clark used to play for the Pittsburgh Steelers, went to Denver to play a game and suffered consequences because even though he doesn't have sickle cell anemia, he has sickle cell anemia trait, which means he's heterozygous basically. And it doesn't affect him, but in Denver being a mile high, it did affect him. So folks with sickle cell anemia trait have that problem. Now, people with sickle cell anemia have a whole bunch of problems, like we were talking about, in genetic mutations. Remember, sickle cell anemia is one of those pleiotropic traits where you get one gene, holy cow, one base here, and you get all these effects. Something interesting about folks with sickle cell anemia or sickle cell anemia trait, you're actually naturally immune to malaria, which explains a lot of things about the disease. And if you want to know more about that, you can take my Bio 2 class and we do talk about that. All right, I got one more slide. Let's look at other types of mutations. So again, up at the top, we have the wild type. This is the way it's supposed to be, the top. And what we're going to do is going to take that wild type, we're going to do some changes with it. First, let's do a point mutation. But let's do a point mutation to a wobbly base. Let's do a point mutation to the third base in a codon. See? Instead of an adenine, you get a guanine. And what you get there, instead of a uracil, you get a cytosine. So what happens is that base that's U is actually the one that's being modified. But since it's a wobbly base, it doesn't matter, so we get a silent mutation. If, on the other hand, it's not one of those wobbly bases and you get the wrong amino acid, you get a missense. Is that a problem? Well, that depends on how bad it is, right? That's what I mean about sickle cell anemia. It depends on whether it's hydrophilic for hydrophobic, or it depends on how very different the amino acids are. And then if you get a mess up in one of those first bases in the second codon, you don't even get an amino acid or a protein. You get an amino acid. That's it. You just get methionine. You get nonsense. These over here on the right, I always think about these can be particularly a problem. They're called frame shift mutations. And to me, frame shift mutations are kind of like if you misbutton your shirt. What I mean by this, if an extra base gets added or a base is missing, so this is added or missing, what happens is when the ribosome reads the piece of messenger RNA, see, this mutation occurs on the DNA. So when the ribosome reads the piece of messenger RNA, it obviously doesn't know the mutation has happened, and it knows it reads codons 3 at a time. So if there's an extra one in there, you could get, like at the top one you see there, you could have a start codon AUG, and then because of an extra adenine, you get UAA, which is a stop codon. So you don't get anything, right? That would be a nonsense again. The second type of frame shift mutation, man, that can be a problem. In the second type of frame shift mutation, it isn't one of the wobbly bases necessarily, but what it does is it affects that amino acid and everyone downstream. See, leucine and alanine, all those are different. So that's what I mean about you have your shirt misbutton. You know, if you mess up that first button on your shirt, then all the way down to the very end, they're going to be messed up. And in the last one, that's not a frameshift mutation, but a whole codon could be missing, or a whole, actually not a codon, but a whole 3 bases in the DNA can be missing, which means you're missing an entire amino acid. Now, again, the effects of all these, silent mutations have no effects. We probably all have silent mutations. But the effects of all of these vary on, what gene is being affected and how badly it's being affected. I guess I would say probably some of the worst would be the nonsense mutations, because you don't get a protein at all. Of course, that may not be a bad thing. That could be why, example, there are purple and white flowers in Mendel's flowers. A frameshift mutation like that, again, the same idea. You get no protein at all. I guess these would be some of the worst, I would think. I mean, because you're messing up start codons, stop codons, you're messing up letters all down the road. It's like I said, messing up your shirt. And again, whether it's a different amino acid or one's missing, it kind of depends on which one it is. So you might have some of these in you, and yet they're not significant enough to cause a problem. If you remember, enzymes and substrates even have that induced fit. So if the enzyme doesn't have exactly the right shape, say it's a little bit too thick in that one area because of maybe something like this, maybe missing an amino acid, so the folding isn't quite tight enough. Well, the enzyme and the substrate, it's that induced fit. They said they're close enough to let them fit. Mother Nature always provides we're playing with probability here. Not every time are these molecules going to act the exact same way. So she gives a little bit of a leeway on the other side, right? Okay, folks, any questions for me? I'm going to stop the recording.